morning, everyone. It is my pleasure to introduce today's speaker, Dr. Gaurav Singh. Dr. Gaurav is a Research Associate Fellow at the Institute for Stem Cell Science and Regenerative Medicine, inStem, at Bangalore. He earned his PhD in nanoscale science at the University of North Carolina at Charlotte, where he developed multipulse ultrafast fluorescence spectroscopy to dissect excited state dynamics in colloidal nanocrystals. Since moving to cell biology, Dr. Gaurav has combined advanced fluorescence lifetime imaging FLIM with molecular proto-based biosensors and biochemical and genetic assays to investigate how mitochondrial membrane organization regulates respiration and redox signaling. This work led to two complementary studies, one showing that cells modulate inner membrane fluidity to match respiratory demand, and another revealing that cytosolic reactive oxygen species, rather than respiration, rapidly reshape mitochondrial networks. Dr. Gaurav is passionate about developing next generation FLIM technologies that would allow us to measure biochemical function at nanoscale in living cells and tissues. So with that brief introduction, I invite Dr. Gaurav to deliver his talk on the multiphoton imaging biology opportunities and challenges. Over to you.
Thank you for that wonderful introduction. Thank you, Nehal, and for inviting me, and Sandhya for really organizing this trip. So my name is Gaurav. I work at inStem, Bangalore, with Dr. Sunil Lakshman. And so I'm going to talk about something. If you feel the words are a bit jargonish, there's too much technicality, feel free to interrupt me, ask me. And so I wasn't sure about the audience. So there might be doctors, there might be pathologists, there might be different kinds of people. So I've tried to keep it a bit general. So if you have specific questions, you can stop me and ask, or you can just talk to me after the talk. I'll be happy to answer. So again, I'm an engineer by training. I dabbled with chemistry and photophysics of nanoscale materials for my PhD. And then I moved to biology, where I've been mostly using different kind of imaging tools to answer questions in cell biology, mostly around mitochondria. How does mitochondrial membrane changes, can we measure it, and can we relate it to the functions of the cells? And maybe we can use that for diagnostic purposes, or maybe understand disease and dysfunction in cells and tissues. And so mostly, so I'll talk about these images later, because they'll come in the context of metabolic imaging in cells. But to give a short summary, that I mostly work with cells, not so much with tissues or live animals. And we also work with developing a lot of probes, so that can go and stay in the cell. So all this sort of squiggly things which you're seeing are mitochondria in the cells, and all these are live imaging. And the color which is actually the fluorescence lifetime of that probe. And the color actually tells you the fluidity of the membrane, mitochondrial membrane. And mitochondrial membrane is the place where respiration happens. So by using these probes, you can say something about the organelle membrane in live cells and measure the function. And we showed that in a study which we published in 2023, that the fluidity of these membranes is key in determining the respiration status. So if the cells are respiring more, the membranes are more fluid. And we measure that again using a complementary NADH autofluorescence FLIM, which I'll talk about. And we also try to increase the resolution of our imaging. So we also develop probes, which we can also resolve the internal structures of these tubular organelles. So if you kind of zoom in into these organelles, you see that the membranes of these organelles, the internal membrane is actually folded. And this is the structure you usually see in electron microscopy. So we can also see that in high resolution optical microscopy. Again, we also are interested in looking at the shape of these organelles. How does that change? So for example, in these two cases, in the cell, the mitochondria is more tubular, more networked. In this case, it is more fragmented. And we showed that actually it's a reactive oxygen species inside the cells, levels of these ROS, which actually determine the shape of this network. And then again, we're thinking about how can we use this for sort of diagnostic purposes. If you see a fragmented mitochondria, can you say something about the oxidative status of the cell or the tissue, stuff like that. So today I'm going to talk about multiphoton. And before I begin, what exactly do we mean by multiphoton, right? What is one photon versus multiphoton? And so we'll begin with a basic sort of a light absorption process. So for example, if you had a molecule, right? So you have a molecule, you shine light, right? What happens? Some of the light will, if the molecule is a little bit transparent, will pass through. Some of it will be absorbed, right? A lot of it would be scattered back. That's how we see things. Light is reflected back. But there are special molecules called fluorophores, which can actually absorb some of this light, take some time. So they'll absorb the light. So, and then that light is used by the molecule to get excited. So the molecule is promoted to the excited state. And so this process is called light absorption. The excited state stays for a little while, and that little while could be few nanoseconds, mostly. In some molecules, it could be tens of nanoseconds. In some molecules, it could actually be hundreds of nanoseconds, even microseconds. And after spending some time in the excited state, the natural tendency of the molecule is to come back to the ground state. And when it does, it can release that energy by emitting a photon, and you see fluorescence. And usually this fluorescence is redshifted. So that's why all your probes, so you excite slightly in the bluish or a shorter wavelengths, and the emission or the fluorescence is usually in the longer wavelengths. So this canonical sort of a fluorescence absorbance fluorescence process in most normal cases is actually a one photon absorption. So when you shine light, the probability of absorbing more than one photon simultaneously is almost close to zero in your normal imaging conditions. And so this is actually the key. So now many people confuse that multiphoton is about near infrared imaging. That is not true. So these days we have fluorophores which can absorb all the way from UV. You have calcium dyes which absorb around 350 nanometers, right? You have DAPI which absorb around 350. You have visible fluorophores, but you also have fluorophores that can absorb near infrared radiation. And in that case, that imaging is also one photon fluorescence imaging. That is not multiphoton imaging, right? Because it's still absorbing near infrared one photon light, and then you fluoresce that. So that's your normal. So all your normal fluorescence imaging is one photon imaging, right? Now what happens in that special case when you really shine an intense beam of light of a longer wavelength? So for example, your molecule have an absorbance around 400 nanometers, right? Now clearly, that's the energy difference to promote the molecule from ground state to excited state, right? So if you use longer wavelengths or a lower energy, you can't promote the molecule from ground state to the excited state, right? So the absorption won't happen. And which is true, right? So if you have a dye which absorbs in the visible range, right? You shine red light, nothing's gonna happen, right? But you keep on increasing the intensity of the light, right? Eventually, there would be moments when the molecule can absorb two photons at the same time, right? So what it can do is it can absorb that 800 nanometer photons, and it can then move from ground state to the excited state, and then come back to the ground state by emitting a photon, just as you would have done in the normal case. So this is called two photon absorption, which means you're absorbing two photons simultaneously. So I told you, right, it's a very rare process. It won't happen normally. And if you really use very high intensity to induce or to increase the probability of having such a high number of photons in your beam that you can absorb two photons, you're going to burn your sample, right? So what do we do? So we figured it out that how do we increase the probability of the molecule absorbing two photons at the same time? And the trick is actually using pulse lasers. And so what are pulse lasers? These are lasers which are pulse, which means they send off their energy in bunches. So for example, I've drawn this, I'll come to it. And the advantage is that these pulses can be made very short. And how short? This can be as short as 100 femtoseconds. So when you do that, you're actually condensing all that intensity in that 100 femtosecond time window. And when you do that, you really increase the probability of that molecule to absorb 2 photons in that pulse. And so you can then absorb 2 photons, get excited, and then come back to the ground state by emitting a photon. So that's why all your multiphoton lasers are essentially your ultra-fast pulse lasers. So one, you could say, limitation or specification or requirement for doing any multiphoton imaging is that you need lasers at a pulse, and the pulses have to be very short, at least 100 to 150 femtoseconds, more or less. So it's just a light pulse I've shown with a pulse width. And this might seem like a limitation. Actually, it is one of the challenges that the instrumentation becomes complicated, complex, and costly. But one advantage it inherently gives, because you're using a pulse laser, you could also do other kind of imaging. And the other kind of imaging I mean is the fluorescence imaging, fluorescence lifetime imaging. And I'm going to talk about it a little later. And what I mean is that when you shine light on a molecule, the molecule absorbs, the molecule emits fluorescence. But I told you, it takes a certain amount of time, right? It's not an instantaneous process. The molecule sits in the excited state. And the time it sits in the excited state is the average fluorescence lifetime of the molecule, right? For some molecules, it could be short. For some molecules, it could be long. And we and others have shown that we can actually use this property to say something about the environment of the molecule. And if these molecules are in cells and tissues, we can say something about the state of the cells and the state of the tissues. So we can use this modality of fluorescence lifetimes to actually do basic biology, diagnostics, and many different things. And the fact that all multiphoton lasers are pulse lasers, so they are inherently compatible to do fluorescence lifetime imaging. So that's one advantage that comes right off the package when you do multiphoton imaging. So now, what are the distinct advantages of multiphoton versus a one photon imaging? And the first advantage, which is actually the key to the whole process, is this inherent sectioning capability. So, for example, if you had a cuvette, so actually you had a cuvette, it's filled with a fluorescent dye. Sure.
How does this laser differ from therapeutic lasers given for vascular proliferation, etc.?
So that's a good.
Sure.
So
how does the laser that you use here differ from the lasers which are utilized for therapeutic conditions, like, for example, vascular proliferation and retinal disease, etc.? Is there anything in terms of intensity or
I would assume some combination of the wavelength and the intensity, where either you want to induce some sort of a damage or so I think it's a combination of that. But I won't have much information because I'm completely out of that field. But I'm assuming it would be something like that. So again, so you have to be careful. Like if you really keep on shining that intense multiphoton beam, you'll end up burning holes. So again, by controlling the intensity, the power, the wavelength, and the duration, you can actually sort of decide what do you want to do at a particular point in the sample. So all imaging, I mean, is in a way is destructive. There's nothing called non-destructive imaging. So all fluorescence imaging, even in live cells and tissues, you're affecting the sample in certain way. So so one sort of a advantage which is really sort of at the heart of multiphoton imaging is this inherent sectioning capability. And what I mean is that anybody who has done fluorescence imaging, right? Let's say if you have a cuvette full of sample, you shine light, you know that the focus is not a sharp point, right? there's always this sort of a convex sort of a tail in the zone which actually is there, right? And that's the reason we do confocal imaging, right? So we want to remove this out of focus excitation in our samples, right? So that's why we move from wide field fluorescence imaging to a confocal fluorescence imaging. But if you do a multiphoton excitation, so I told you, right, the absorption really depends on the probability of absorbing more than one photon, right? And that probability is highest when the intensity is highest, right? Because it really is critically dependent on the intensity. And the beam is most intense at the focal point. So in the multiphoton absorption, the absorption only happens very close to the focal point. So if you see your excitation, it's very close to a point excitation. So you have no out of focus excitation. And now that's very advantageous because you don't have to use a pinhole, right? You don't have to do confocal multiphoton imaging to do your sectioning. So now you can actually sort of image the whole depth of your sample with a very good signal to noise ratio. Because when you use pinholes in your confocal microscopy, you do remove a lot of out of focus light, but you also lose a lot of signal, right? So there's always a trade-off. So that's one advantage that really sort of comes right off the bat. You have localized excitation. You don't need a pinhole. It's inherently there to do your sectioning. And if you combine that inherent sectioning capability with the ability of this near infrared radiation to penetrate deeper into the tissue, you actually then unlock the biggest sort of the selling point of multiphoton excitation is deep tissue imaging. So again, I don't do any deep tissue imaging. So I've taken this off from a review article. But the basic point is that, and we do that actually, we do that in LCBS. A lot of my colleagues do that. And you can actually do live brain activity imaging in rats, right? You can do a surgery. You can put a cover slip window on the skull of the rat, and there you can make it stationary and all that. And the rat, either you can inject a probe, you can inject a lot of probes, or you could have a transgenic mice, right? The mice is expressing fluorescent proteins in the brain or in the neurons. And then you can actually image neurons really deep. So this is, I think, almost close to 1 millimeter. So you can really map brain activity in living animals in live tissues really deep, because you have that inherent sectioning capability. The wavelengths can go deeper into your tissue, into your animal, and they can excite really deep, and then you can collect your signal from deep tissue. Another advantage that really comes handy with multiphoton excitation is, so again, so you have to also keep the physics in mind. That's why I told you, right? It's one photon versus two photons or multiple photons absorption. So it's fundamentally different, right? And another fundamental difference is this. There's something called absorption cross section. So we'll not go into the technicalities, but the take home message is that the two photon absorption is a little bit different. So many molecules will have a different absorption spectra, right? So you usually excite them at different wavelengths. But in the case of two photon excitation, many molecules can be excited with a single wavelength, because most molecules have a very high two photon absorption cross section in the around the 400 to 450 nanometer range. So you could have molecules which otherwise you need, for example, green or red light to excite, or blue light to excite, or green light to excite, but they all can be excited by the single 800 nanometer excitation. And this is really advantageous, because in many cases you don't want to change your excitation wavelength, because when you change your excitation wavelength, right, everything changes, because the refractive index of the sample depends on your excitation wavelength, your spherical aberration, your chromatic aberration, that depends on the wavelength, right? So there would be a lot of slight changes in the focus, the way you focus, right? So there will be the focusing is blurred, there will be halo in the sample. So the fact that you can excite multiple fluorophores with a single wavelength is really very, very advantageous. You're doing complicated samples.
So, Gaurav, is this for multiphoton? What I have is this for a specific homogeneous specific molecules for the multiphoton with the single exactly?
So, so partially no. So pretty much all commonly available fluorophores, especially small molecule fluorophores, they can be excited very efficiently at 800 nanometers. Even though if you look at their one photon absorption, they might have a very poor one photon absorption, for example, at 400. But when you excite at 800, they'll absorb that 800 nanometer. And that, again, so that's so. So when we talk about two photon absorption, we have to keep in mind that we can't really think from the perspective of a normal one photon absorption. So, so in the ideal scenario, if you, for example, if you're passionate about doing some really specific two photon absorption with your molecules, the molecules or labels, fluorescent labels which you have designed, so you actually have to measure a two photon absorption cross section to find the most optimum wavelength. But usually, most sort of commonly available fluorophores are very good. They could be excited at 800 nanometers. So actually, we did a sample yesterday which had, I think, blue, green, red fluorophores, and they were very happily lit up with the 800 nanometer light. So now there's three advantages, right? One is that those wavelengths allow us to penetrate deeper. There's inherent sectioning capability, right? So you don't need any pinholes, anything. You can just simply go and start sectioning your sample. And the third is the ability to excite multiple labels with a single wavelength that makes the whole imaging excitation acquisition process very easy and straightforward. And then that really unlocks all the different imaging modalities then you can do, right? Again, this is none of my domain, but very good reviews, and essentially people have been able to look at live captures of microbes by macrophages in so this was, I think, done in live animal. People have looked at blood vessels and how does immune cells come around those blood vessels. People have looked at in the cancer tissues. I think this is some sort of bone tissue or something where people have looked at vasculature, bone, and how does other cells come and all this is in live animals or live tissues. Because again, you have this capability to penetrate deeper, to section, and then to excite multiple labels at the same time. So you can do all this. So there's a whole sort of a field where you do deep tissue in vivo microscopy. People also call it, I think, intravital, multiphoton intravital microscopy, which you can do. Any questions till this part?
How good was the reliability of the tissue and
So as I told you, there's nothing called non-destructive imaging. But I did saw that many people actually had done time-dependent imaging. So they could sort of adjust the dose of their excitation, and they could image at the same region within, say, 1 week or something. So again, so you're absolutely right. Like when you do such kind of intense imaging, there could be local damages, which again could affect what you're looking at. So you have to be very, very careful.
specifically in terms of like the resolution? Because when you're going 500, 1000 microns, that's quite a bit of a thickness. As far as section tissues are concerned, do you think we have to process a particular sample for a photon? The advantage being you can have good resolution, but the quality of the resolution, you can see those individual
So that's a good question. So I'll flip this question in this way, right? So we're living in an age where, for example, if you cut a tissue, right? It's a thick tissue. Let's say it's a 200 micron thick tissue, and you fix it, right? And it's labeled. It already has a different part labeled with probes. You go and do a confocal imaging, right? You'll penetrate a certain depth. You do a multiphoton imaging, maybe you'll be able to penetrate a bit deeper. But we also have this methods now that you could do these clarity methods where actually you can make the sample transparent, right? Now the question is, do I still need to use multiphoton in those cases where actually I can use clarity and make my sample transparent, and then I can go back and simply use the single excitation confocal imaging. And then, so that's a call which you have to take, you know, is the sample processing using other methods would change the sample vis-a-vis how much depth and resolution is acceptable to you. So I feel like there are trade-offs in all these methods, and like you have to take a call. So it's very hard to give you like give sort of an objective answer that this would be the best. So you're absolutely right. So I told 500 microns to 1 millimeter, right? But those would be very few labs in the world which are doing deep tissue 1 millimeter imaging on a very routine basis, right? Most of us, if we are lucky, a few hundreds of microns, I think you should be happy. So practical resolution in the systems, usually we have the commercial systems, I would say 300 microns, something like that. So, but again, so you can go up till one. People claim 2 millimeter also, but again, I don't know how much. So again, that also depends on the tissue, right? Is the tissue more scattery? Is the tissue more transparent? So it really depends on a sample preparation. Really, I mean, most of the imaging, including 2-photon imaging, really depends on the kind of the sample you have and how much creative you are in processing that sample, right? So if you can't see through the sample, right, there's a lot of things which are scattering. I mean, 2-photon microscope is also not magical, right? It will not magically penetrate and show you anything. So absolutely right. That's a very good question.
Yes, sir. That is complex.
I mean, it's really push up the cost significantly. How cost effective?
So I think these dyes are fairly not that costly. Yeah, very reasonable. So there are dyes which people use to label blood vessels and many other things. So I think very reasonable, very routine. Less expensive probably than the facility cost you have to pay like for 1 hour charge or something. I don't know how much is that there, but not that expensive. Not that expensive. Again, so again, it also depends on the question you are asking, right? For example, if you're doing antibodies, right, to label something, and those antibodies are really expensive, so that would push up the cost. So again, it depends on many other factors. So again, any deep tissue imaging is not magical. It really depends. So it's a very good question. So I would urge anybody who wants to venture into multiphoton or even normal imaging, really pay a lot of attention in thinking about your samples, right? So until now, so I talked about multiphoton deep tissue imaging, right? But everything was labeled, right? There were labels, the cells were labeled, the tissues were labeled, they were excited by our multiphoton excitation, right? Sure.
So, tell the when should one?
That's what I was explaining, right? So
when I do a lot of normal tissue. So is it going to be because it is going to preserve this tissue?
so okay. So there could be two ways of answering this question, right? A lot of people section their tissues, right? They make thin slices. They make like 50 micron slices or 20 micron slices. I don't know. You can go and do your confocal imaging. You get very high signal to noise ratio. You get really good contrast, right? I don't feel multiphoton imaging there would really make a dent. But there are exceptions. So I'll actually come that where exactly can you specifically use multiphoton versus one photon excitation, even in normal circumstances. But let's say, so the biggest advantage I feel, or the biggest use of multiphoton excitation comes in live tissues, live thick tissues, live animals, right? If you're looking at, so I think that's where the game is. That's where you need to push the boundaries. Like, why not look at cancer cells or tissue or tumor growing in the animal, right? Why not look at the dynamics over there? You can use multiphoton there.
The type of fluorophores which are used here, are they different?
Exactly same.
Same thing.
Nothing changes. In principle, in principle, right? But again, so in most cases, you'll just do a hit and a trial, right? Which is the most optimum excitation for your sample.
So that was my question. There. So in that case, I would not know whether confocal or multifocal, which is going to get better result. Should I try both and then decide?
So in thin samples, invariably, confocal would give you better results. Invariably, right? See, also you have to keep in mind, so I'm not going to go into the technicalities, how actually we do multiphoton imaging. There's a little difference where the detector is placed. So in your confocal imaging, what you do is you do D-scan imaging, right? Both the excitation and the emission passes through the scanning mirrors. In the multiphoton, you do something called non-D-scan detection, right? So the detection is collected right off after the objective. So you also collect a lot of background. So you have to really make sure that your room is dark, your screen, even your laptop or your computer screen is off. There's no lights or it's very sensitive that way if you're doing a particular kind of non-D-scan multiphoton detection. So it really depends. But as a rule of thumb, for thin samples, thin fixed samples, usually I would prefer confocal. But I'll come to the specific places where you can actually do that. So again, all these were labeled imaging, right? We just use a different kind of excitation to excite multiple fluorophores, maybe, or push deeper and do many things and do sectioning without using a pinhole. The place which is really exciting is called label-free imaging. And a lot of people want to use, multiphoton imaging without adding any labels. Is it doable? And yes and no. And there are many ways you can do actually label-free imaging. The first is something called scattering, right? Which is second harmonic generation. And this is so this is my own sample. This is not my sample. I was working with a collaborator who works on skin biology. And this is actually a skin section, I think probably somewhere around 20 to 50 microns. And you can see these hair stems in this skin. And now what is second harmonic scattering? So everybody knows scattering though, right? Understand scattering? So if I shine light, some of that light would be scattered back, right? At the same wavelength, right? So if I shine 400 nanometer, 400 nanometer would be scattered back. Same thing happens also in a 2-photon excitation scenario. For example, I excited, say, 800 nanometer light, right? So two 800 nanometer photons could be absorbed, will be equivalent to a 400 nanometer excitation, right? And if it was a fluorophore, it would have absorbed that and would have given us fluorescence at some longer wavelength. But many times what happens is that if you have some sort of an ordered structure in the cell, for example, if you have these collagen fibers, or you have other kind of ordered structures in the cell, those structures essentially acts like a diffraction grating. They can actually scatter the 2-photon light. So you can then absorb those 2 photons, essentially convert them, in a very loose sense, into 400 nanometer, and then scatter off that 400 nanometer light. So your scattering is half the wavelength, right? And that scattering is very intense. And people like us and many other people have actually used that scattering to look at collagen biology. So for example, in this skin tissue, you can see this sort of bluish color. This is all collagen, which is doing second harmonic scattering. So if you have tissues where you are interested in looking at the collagen, that really gives a very nice second harmonic scattering signal. So you can use your multiphoton excitation to do deep tissue collagen imaging. People have also figured out by measuring the, so I told you, right? One advantage that comes with multiphoton is the ability to also measure lifetime because it's a pulse laser, right? So people have then done multiphoton fluorescence lifetime imaging and then use different lifetime signatures to look at different types of collagen. So people have done that. So it's very sort of a productive field. And so one is scattering, right? So that's again a label-free contrast. You can look at collagen, and there are other structures where people have also looked at, right? So mostly, if you have an order structure in your tissue or in your microenvironment, you could use these kind of scattering signals. Second is autofluorescence. So in this case, since I had sort of these hair stems, there's a lot of keratin, so I have very strong autofluorescence. So you can see that actually. These are sort of hair follicles are different in color. Now, the problem with autofluorescence imaging is it's very hard to tell what is autofluorescing, right? So you have so many different things in your cells and tissues, right? Which could be giving you autofluorescent signal. So you can look at this one way that you don't care what's the origin of that signal, right? If you can excite your sample and you can correlate that signal with a known feature in your sample, then you can use that to do label-free detection or identification of features in your tissue. So there's one way of looking at it. Another way is that you would ideally want to know what is there, what's a species which is fluorescing, right? And by measuring the autofluorescence, can you say something about the state of the cell or the tissue? And you can. And that third kind of sort of a label-free imaging is something called metabolic imaging. And what people have done is that, we have, okay, everybody knows NADH and NAD plus, right? Little biochemistry. It doesn't matter. It doesn't matter. So you have these metabolites, which are very important, right? And if you have studied glycolysis and respiration, you know that the ratio of these metabolites will change, right? The redox ratio will change. They'll become either more oxidized or reduced, right? So if you're doing glycolysis, you're producing a lot of NADH, right? If you're doing respiration, you're consuming a lot of that NADH to convert into NAD+. And people realized long back that this molecule, this NADH molecule, which is sort of at the heart of metabolism, if you can measure the ratio, you can say something about the metabolism of the cell, whether the cell is glycolytic, whether the cell is respiratory, and vice versa, tissue also. Whether the tissue is glycolytic, tissue is respiratory, maybe in a certain disease condition, the respiratory status of the tissue has changed. Maybe if the cells are becoming cancer, they're becoming more glycolytic, right? So you can measure that. And people realize that these molecules have an autofluorescence signal, which you can measure. And they also have a associated lifetime that you can measure. And by measuring either the intensity or the average lifetime of this molecule, you can say something about the metabolic status of the cell. So, of course, you can do this in tissues, but you can also do this in the cells, right? So, and that's what we did in our case. So we had these fibroblasts, right? These are live cells. So we're doing live imaging. And so one advantage I told you with the label-free imaging is then you have one label-free, then you can also start adding labels, right? So it really makes it very easy. So we had the cells labeled with our probe, right? This is a probe that goes to the mitochondria and tells something about the membrane. So the color here indicates a lifetime, right? If it's the blue color, it means low lifetime. If it's a reddish color, it means high lifetime. It doesn't matter. Just look at the color, blue or red, right? So blue here means the cells are more fluid. The mitochondrial, not the cells, the mitochondrial membrane in the cells are more fluid. So this is a probe which we made in our lab. We call it microrotor 1. It's a molecular rotor. So depending on the micro environment, the probe will twist. So if it is a more viscous environment, the probe will be planar. If it's a more fluid environment, the probe will twist and the signal and the lifetime will change. So it
configuration to the enzyme.
so our model is that it goes and assembles in the membrane. It doesn't really attach to any particular enzyme or lipid species. It's mostly a charge-based interaction because this probe is positively charged and mitochondrial membranes are negatively charged. So they accumulate a lot of this probe. So to best of our knowledge, it's not really specific to a enzyme. So it really sort of stains the.
But that itself would have gone through a huge process of validation.
Of course.
Particular
Yeah. So I'm not going into all those experiments, like how it happened. It took. So the probe was made by a postdoc, a couple of postdocs synthesized and then identified, and then I pushed that work to find where the probe goes, what is measuring. So yeah, it did took a lot of.
So Autoflow is it has like specifically if I'm working with the cell that so it has its own pros and cons. I think for you it's helping, but for multiphoton, if I'm going deep imaging, at times that creates a problem. So just on a just to ask you on that, can you metabolically deprive cells in order to minimize autofluorescence, considering it is a metabolically.
So the problem then is what are you measuring, right? Because let's say if you want to measure a metabolic state and you deprive.
Of course, we play around with various components and cell culture components.
So you can't do that. So people again, so I should before I so the reason autofluorescence is because it is the intrinsic fluorescence of the cells, but we also have to keep in mind that the autofluorescence in most cases is very weak. So if you have an external label, that would any day, if the label is in sufficient quantity, again in double quotes, if you have sufficiently labeled your cell or tissue, it will any day dominate over autofluorescence. But there would be cases, right? There would be a certain cell or a tissue type where certain concentration of pigments or metabolites might give you a really strong autofluorescence signal. And my advice in that case would be change your label, like move out of that autofluorescent window, maybe use a redder label that's not interfering with it, or use a brighter label rather than chasing that sort of road to minimize autofluorescence, because that's a very dark alley, would lead nowhere in my experience. And also, again, there are optical techniques, some of which I worked on in my PhD, which you can use to minimize autofluorescence. But again, that's very niche and technical. Like maybe some other day I can discuss with you. But yeah. But the point was that again, we were interested that if you have the mitochondrial membrane in a certain state, is it more fluid or ordered? What's the respiration status of the mitochondria, right? Because we wanted to correlate that physical quantity with the respiration status. But how do we measure respiration, right? In live cells. Can we say something about the metabolic status? And we can. So we did the NADH flame. We measured the average lifetime. And you can see that this is right. And we did a lot of control experiments to show that we are really measuring what we are measuring. That cells which are more fluid are actually more respiratory. And there is an inverse relationship. We published this paper. So again, in this particular study, we were able to do a label-free metabolic imaging to say something about the metabolic status of the cells. And we excited the sample at 720 nanometers to do our multiphoton NADH imaging. And our dyes were excited at 800 nanometers. And then we collected that in 2 different channels. And we were able to do this simultaneously, right, in live cells. So I am assuming in certain cases, it might be even possible to do in live tissues. And people have done that. So this is a paper from, I think, Daniel Needleman's lab at Harvard. And what they have shown, I think these are mouse oocytes, if I remember correctly. And they looked at the NADH autofluorescence and they varied the amount of oxygen in their media. And they hypothesized that if you had more oxygen, the mitochondrial respiration should be higher. It should be induced. And they see that the NADH autofluorescence dramatically goes up. And they also did some staining to show that all of this NADH was actually metabolizing in mitochondria. So you can really sort of measure compartmentalized metabolic activity, at least in oocytes, right? And maybe in other tissues as well. we worked with a lab in NCBS to look into blastocyst, like embryos, and we were able to show that if you have a mitochondrial, by imaging NADH, you can say something about whether the mitochondria or the mitochondrial respirations in that embryo is compromised or not compromised, if they're glycolytic or less glycolytic. So you can do these kind of experiments. I think
It's like you're using label-free image. You're like exiting the previous pattern.
Correct.
So will that affect the medical state of the cells?
so again, there's a, as I told, there's a
So we are using NADH, let's say the molecule to raise the medical. We are exiting these molecules, will it affect the metabolism?
so let me rephrase this. Essentially, you're asking that does the act of imaging these endogenous metabolites will affect the overall metabolism? And answer again, this is so the answer is yes and no. So in principle, it should not, because the fluorescence is a very fast activity, right? So the molecules absorb light very fast, and they this fluorescence is on the level of a nanosecond. So you haven't really perturbed the electronic state of the molecule to behave differently in its niche. But again, when you excite any molecule, there is a chance that some molecules would be, you can permanently oxidize them, right? And so maybe if you do long-term imaging, you might affect it a little bit. So it's very context dependent.
I think you have to do the validation experiments to answer that question.
Yeah.
So for example, if you have a mitochondrial disease and you add this, you may get a blank over here. It won't show up or it will be much less. And that, of course, is something which we'd like to get. If I like to treat the mitochondrial disease, I'd like to see the response. So I suppose you need to do a lot of control experiments with that particular tissue and what we consider to be normative. And only then start using it in various disease states. And
correct.
So it's very organ specific.
Yeah. 100%. And also, as Gaurav himself was saying, that it also depends the kind of tissue you are looking at. Some tissues really have a very high intrinsic autofluorescence in that particular range. It might blur our metabolic signals from these particular metabolites like NADH or FAD. So again, it's very context dependent. You really have to do. And so, for example, if you are looking at this, so what I would do, the kind of controls I would do would be that I'll add metabolites or drugs or chemicals to either induce mitochondrial respiration or to inhibit mitochondrial respiration, and see then what happens to my signal, right? So if my signal changes as a function of that, I'll be a little bit more confident that my signal is really telling me something about the metabolic state of the cell. So yeah, so there has to be a lot of controls. So again, as I was saying that we were doing some NADH imaging of blastocyst, these embryos. But the point I want to make is that if you want to really do metabolic,
Why is the central area all black? Shouldn't that be metabolically active?
So I think this is like an early stage embryo, something like that, and maybe we didn't have much NADH signal from the center of the blastocyst. So possibly that. So again, it's a very crude, like one sample image. So I wouldn't want. Possibly. So I think this was live. This was a full embryo, a full blastocyst, whatever that is. So this was like an intact sample. This wasn't a slice. So this was live sample. So we were interested in. So we had a person who was interested in. So again, if you have tissues, right? How do you look at mitochondria? I mean, the dyes don't penetrate that much, right? And you want to look at the metabolic state. How do you do that? You can't do oxygen consumption of the cells inside the tissue. So one way of doing that is that looking at these metabolic ratios. So but people have done that. People have done that in embryos. Like there's a lot of now review articles where people are planning to also use this to. Oh my bad! I think I stopped. People have looked at this to look at the quality of the embryos, whether it is metabolically in a better state or in a worse state. So really, the field has really advanced. So again, I thought that if you shining such an intense light on that embryo, is it really feasible to use that embryo? But people have shown that it's very viable. And so again, the technology is really pushing the frontiers, and you really have to play with sort of different parameters to make it happen.
Right. So does it mean that the signal is from the whole, each section of the embryo is broken?
So again, this is a very good question. So we didn't have a very optimized setup then, and we were actually, so I looked at the most focused plane in the sample. So ideally, I should have done a full Z-stack imaging, which I didn't do. So then the picture would have been a little different. As Nihal also asked, like this looks suspiciously empty in the center. So this is a very crude single plane focused imaging. So clearly the embryo was a little thicker, so not a very representative, but just to give you sort of a flavor that you have a contrast which is label free and which says something, and you can use that in different contexts. So, so one thing I would say, people who want to do sort of metabolic label free imaging, of course, you can do intensity as David Needleman's lab showed in intact in live mouse oocytes that you can do that, and NADH intensity is in itself a sufficient signal. But if you really want to do it, I would say combine it with fluorescence lifetime imaging, because fluorescence lifetimes actually help you identify that is this really so. So, for example, there could be multiple autofluorescent species, right? How do you distinguish them? Actually, one way to do that is fluorescence lifetimes. Many different molecules, they might emit in the same spectral region, right? So maybe uh, me and some other molecule, we are both emitting, say, in the green region, but we both might have very different lifetimes. And by measuring the lifetime, I am able to distinguish whether this is molecule A or this is molecule B, even though they might be emitting the same emission window. So that lifetime then give you another sort of level of contrast to make a judgment that is this really NADH or is this some other thing which is interfering. So people who do really serious metabolic imaging usually combine it with fluorescence lifetimes to really make sense of this whole business. And I told you, right, multiphoton imaging is inherently compatible to do fluorescence lifetimes because it is a pulse laser, right? So you just put a photon counting card and you are all set to do lifetime imaging with a few. I mean, it's a bit complicated, but in principle, it's very doable. So, so all advantages, but again, there's a lot of challenges, and the most biggest challenge is that it's a costly process, right? You need to buy that expensive laser. You need to add that to your microscope with a specialized optics, a different detector, a different modality. So it's a little costly. And again, you still need contrast, right? So many of you might have thought that, I'll just shine multiphoton light and I'll start seeing things. That's not true. You still need to label your samples, right? You still need to use a transgenic mouse or you add external labels and then go deeper in image, right? But you need labels. And again, if of course you can do label-free imaging, but again, label-free imaging is artifact prone. So you have to do a lot of controls. You have to, if possible, add an additional lifetime contrast to really be absolutely sure that what you are measuring is coming from a NADH to NAD plus ratio. So again, that's very challenging, especially for a person who is not used to doing a lot of imaging, but doable. All this is very doable. If you are familiar with the confocal imaging, you are all set to do multiphoton imaging in principle. And same, again, you have to, same things. There's nothing complicated. If you're making a samples, you have to think from the perspective, your own perspective, right? Is the sample too thick? Is it like too scattering? You do a few tries, you see nothing, you figure out ways to maybe reduce the scattering or do other treatments. But the imaging per se, the collection and all the basics of microscopy remain same. Nothing changes, literally nothing changes. You're using the same fluorophores, you're using the same emission window, you're using the same detectors, practically same gain on the detector, everything remains same. Only the mode of excitation and how your sectioning has fundamentally changed. So in a way, yeah, pick a good review on stuff you want to image and just start imaging. I'm mostly done. So feel free to ask any more questions.
Different wavelength, right? So what are the chances of spillover when we do multi-color imaging? And so
Multicolor normal imaging.
yeah, and also like compared to confocal, we can do up to 7 colors confocal. So how about what are the maximum number of waveforms we can do? In the multi-color?
I mean, imagination. So again, it's a very loaded question. See, so the way we get contrast is by controlling the excitation window. Where do we want to excite? And the emission window, right? So if there are probes which could be excited at different wavelengths and collected at different wavelengths, we can keep on adding probes. But there would be spectral crosstalk. There would be spillover of maybe the excitation window or the emission window. And then again, you have to take a judgment call. Do you want to collect your data and do a post-processing? So we have algorithms which can minimize spectral crosstalk. Many microscopes these days come with those algorithms. Or you want to choose your labeling strategy in such a way that you minimize your spectral crosstalk. So yeah, so there would always be, if you're using too many fluorophores, it's always good to go at an online repository such as FP base and just put your fluorophores, add all your fluorophores, and it shows you the excitation and the emission window, and see if there's a crosstalk. Same for the case of multiphoton. And the disadvantage with multiphoton is, I told you, right, the same excitation can really excite many fluorophores. So you don't have any excitation control that much, but you still have the emission control, right? So you can sort of select your emission window in such a way that you could do multiple fluorophores. So,
Question.
So Gaurav, just following up, I think you might have interacted with the guy who works with the fields, these fluoro molecules, which are degradable, of course. I think he presented an email back in inStem. And following up on that, what I'm thinking is like, is it, say, for example, if you have a beating cardiomyocytes, and I want to do multiphoton various probes. Is it, and of course, I have to take care of the cells after my probing, after my usage. I can, differentiate them for further lineage and so on. So is it, have you come across or do you know something about those molecules which you can make it in the lab, which can be used by the cells metabolically active and done their work and off they go?
so I haven't, maybe I missed that talk, but like, what's the point is that
Probably cells continue to fight and it doesn't disrupt your cell culture rhythm.
those molecules. Yeah, I would probably have not much clue. Not much. Yeah. Sorry about that. So I did image. I don't know if you. So I had a cardiomyocyte image in the beginning of my slide. Beating car. So I did collaborate with Dhan Lab and then from IPSC patient lines.
What do you feel clinical applications of this tool, if any? What are the clinical applications of this tool?
Yeah, so first of all, thanks Gaurav for that very insightful talk and making it interactive right away. So most of the questions I think are clarified. So the experience, I think from his point as well as my point are mostly from the research point of view. But what we can see is how they translate to the clinical side. For example, from my personal experience, we looked at how it was in a live animal model using two-photon microscopy. We were looking at how we could probably reduce the side effect of certain drugs which are currently in clinical practice. The example I would give is, for example, in an MI or a stroke, currently we give maybe an alteplase, tenecteplase for thrombolysis, but the flip side is around 12% of them develop bleeding. as a side effect or a complication, and up to 2% developed intracranial hemorrhage as well. So we made in a live animal model, we induced a laser-induced injury to the endothelium, basically making a clot. And then we gave the same drug, the altiplase, to see its lysing, that we know it happens. But we also tried another protein which would accentuate the effect of altiplase. Therefore, when we saw that, we know that we can actually reduce the dose of altiplase. The other one has really no side effects. Its bleeding propensity is, say, equivalent to what aspirin would. So we know that, when you add that particular drug to your routine drug, you probably can reduce the dose of the current drug in practice and reduce the side effect. So through such animal models, research-based methods, we can actually see probably having some translation to clinical practice and impact in thrombolysis. So that was one example from personal experience, I can say. So it's mostly research-based, but I'm sure it has a strong clinical impact. So
how does it work with fresh biopsy tissue as opposed to live tissue? I will talk about the live tissue. If I take a fresh biopsy, or if I take fresh placenta, and within an hour or an hour and a half, I do something with it. Will it give reasonably good characterization in those situations?
I think it would. So that skin sort of tissue I showed you was actually a fresh biopsy. Like that was done within half an hour to 1 hour, and we found that that does make a difference. So, for example, if you're doing metabolic imaging, a lot of these metabolites, if the tissue is sitting for a long time, they might not be active, or they might just come out of the cells. So it might matter.
So, what's your ballpark time? 2 hours, 3 hours?
So again, it depends on what exactly you want to do. Do you want to look at features, or do you want to measure something? Because looking at features, I would say then fix the sample and then look at the features. If you want to measure something, right, you want to measure something that is happening while the tissue is there, then as soon as possible, I would say. And also, a lot of people have figured out what's the right time window.
For example, perfusion.
Again, I would have no clue because I would be the wrong person. Maybe I think.
perfusion probably should be in the first, again, it depends on the experiment, the tissue in question, but it should be as early as possible. Preparation and the experiment should go fairly immediately.
For example, if you're interested in looking at the metabolic activity in the perfusion based injury, you can use that metabolic NADH imaging, right? If respiration is happening, the signal would change. So, but then you have to look at the sample as soon as possible to really make any significant.
Any more questions from the audience? I believe there are a lot more, maybe PhD students and so on in the group. So if there are any more quick questions. Quickly, could I ask one more question, or maybe just clarify? Photo bleaching and photo damage is a problem with confocal, and it's probably reduced a little bit more or significantly with multiphoton. And when you say fluorescence lifetime imaging, is that totally avoided? Because I understand it is not dependent on the intensity of the lasers or the fluorophores in question. So is it avoided totally, or it's just reduced photo bleaching, photo damage?
So okay, again, there's two parts to it. So the photo bleaching would still happen as you're imaging, but that would not significantly affect your measurements if you're doing fluorescence lifetime. Because as you rightly said, the lifetime doesn't really depend on the intensity of the light or the concentration of the probe.
Yeah.
So in that sense, those measurements are more robust, even if photo bleaching is happening. But again, all in double quotes, right? But much more robust, definitely compared to intensity. So the reason we were able to make sense of those fluidity measurements and correlate it to respiration status was solely because we were able to measure fluorescence lifetimes. Otherwise, the mitochondrial membrane is really, really crowded, actually. It's a very difficult place to make sense of anything because a lot of things are happening and the concentrations change very rapidly.
Right. So Any last questions? We are fairly within our time, so. Okay, if there are no more questions, I think we'll conclude this meeting. And thank you, Dr. Gaurav, for that very interesting talk and making very. Thank you so much.